change my title a little bit here. Let me see the button here.
Sorry about
really,
that.
oh, no problem. Just popped up right in the middle, so I had to get rid of it. So to really fit more with the theme of the hackathon, I really started thinking about what we've been trying to do with B cells is really, in a lot of ways, hacking them to do things that we want. And in particular, I'm going to talk a lot about a project where we essentially hacked B cells to produce an antibody of interest. Oops, sorry, my slides don't want to advance for some reason. So maybe we're not, oh, there we go. So first, I wanted to start off by acknowledging my lab. When I started this project many years ago, it was really just a cartoon idea that I had. And these are the people that will pop up in a moment here that really made this project work. So in blue are the people that are currently in my lab. Underlined are the individuals that contributed to this project. And in particular, two former members of the lab, Howell Moffett and Carson Harms, are the individuals that took my cartoon ideas, improved them, and then made them a reality in more efficiently and better than I actually ever imagined. And we'll get into that a little bit later. Of course, I also need to acknowledge the funders for this project. We were given generous funds from the Hartwell Foundation, from Vir Biotechnology to work on this project, and more recently from the NIH to continue this work, hopefully for the next few years. I think the basic idea of what we were trying to do in this project is conceptually, I think, quite simple and has been done in a lot of other contexts. And the basic question that we set out with here is, can we engineer a protective immune response? And this is an idea that goes back a while and has been used very successfully in engineering different immune responses into tumors, different cancer immunotherapies that people have developed. Here, we wanted to focus on engineering a protective immune response against infection. And on the left here, I have a very simple cartoon of what hopefully happens when you have a successful vaccination. You start with a naive B cell that expresses an antibody able to bind whatever antigen or antigens are within your vaccine. Through a very complicated process of differentiation, proliferation, and maturation, which I won't get into the details of, the end goal is to induce the immune system to produce protective antibodies. And these are really produced by two major subsets long-term. The first is probably what we mostly think about, are antibodies that are secreted into the body and can be measured very readily in the serum and other locations. And these antibodies are produced by plasma cells. There are lots of different flavors of plasma cells with different functions, locations, all these types of things. But on a big picture, what you hope to generate from a vaccine are plasma cells that live for the rest of your life, pump out antibodies at high levels to maintain a high level of protection indefinitely. Now, that's not what always occurs. And I think with most vaccines, you actually have boosters, and those are targeting mostly a different subset of cells that's produced from this initial interaction. And these are called memory B cells. These cells don't secrete antibodies into the body in the steady state, but when you give a booster vaccination or if you get infected, these cells can very rapidly differentiate into additional antibody secreting cells to supplement the level of antibody. So if you've followed any of the vaccine and antibody level conversations over the past couple of years with SARS-CoV-2, what you'll see is you do your initial vaccine, you get a level of antibody that peaks, comes down, and then plateaus. You then boost, that goes up. And that going up is in large part to these memory B cells that were formed with that first injection. Now, this is all, one other component of this is, you'll notice here on my slide, it starts with an antibody that's got a gray end to it, and then changes to one to a green. And what I'm showing here is a cartoon representation of affinity maturation. So the antibody actually changes and improves during this process. So you start with an antibody that binds with a low to moderate affinity, and you hopefully end up with an antibody that binds a really high affinity to the pathogen of interest in your vaccine. Now, when vaccines work, this is great. This is the process that we're looking for. But the reason we started this project is because this isn't always the case. I think a great example of this not being the case is for HIV, where researchers have been trying to develop vaccines to induce protective antibodies really for decades now. And this has been really challenging to develop. And a lot of that has to do with the incredible mutation rate of HIV that just allows it to escape antibodies. But there are a lot of antibodies that can bind and neutralize many diverse HIV strains. So an ideal vaccine would be able to specifically induce these very specific antibodies that you want. But the state of where we are with vaccination, we're just not there to be able to say, I want this exact antibody that binds to this exact portion of HIV to be able to protect against that. I think the same could be said for universal vaccines, for SARS-CoV-2, for influenza. There's many other infections where people have been able to identify ideal protective antibodies, but not yet generate vaccines that can induce these in a way that we want. So here, what we thought we'd do is we would get around that through essentially, hacking the system. What we wanted to do is take out naive B cells from a person, use CRISPR-Cas9 to replace the antibody that these cells have generated randomly with an antibody that we know would be protective, just our ideal best antibodies for whatever pathogen we're interested in. Broadly neutralizing HIV specific antibodies, or broadly neutralizing SARS-CoV-2 antibodies, for example. After that engineering, we would then take these cells in culture, differentiate those to the two cell subtypes that I mentioned before, antibody secreting plasma cells and the antibody expressing memory B cells, and put those back into a person and essentially engineer a protective immune response. When we set out to do this, we had a couple ideas of what an ideal approach might look like. First, it'd be something that was easy and efficient. If you've got to go through a million steps over a 12-month period before you can get these cells back into a person, this isn't very easy and efficient and not really likely to be able to be implemented on any type of scale. A second important component is that we really wanted to eliminate the antibody that the cell expressed naturally. Now, the reason we wanted to do that is these random antibodies, we just don't know anything about them at this point in time. And the risk, the fear here is that the existing antibody could lead to some sort of autoimmune response or some other detrimental response that we don't want here. So we want to eliminate that risk by eliminating that antibody so the cell only produces our antibody of interest. We also wanted to engineer the cells in a way that allowed for normal expression and regulation of the antibody. Antibodies can be modified and changed in lots of ways that are really beneficial to the immune response. We're also really struck by the immense secretion ability of an antibody secreting cell, and didn't think we could do any better than what evolution has done in this regard. So I think the estimates are that a single antibody secreting plasma cell can secrete somewhere between 1,000 and 20,000 antibody molecules per second, which is a stunning rate when you start to just consider how many total proteins a cell usually expresses. These cells really just change their whole function in life to really supporting out and secreting antibodies. So we didn't want to mess with that. And finally, we wanted to develop a system that was protective when vaccination is not an option. Now, I mentioned before situations when vaccination is not an option, when you don't have a protective vaccine for a pathogen. But another situation that we think is equally as important is when people are immunocompromised. So when even if you have the greatest vaccine that works in a healthy person, this vaccine won't work in a person whose immune response is compromised, and they're not able to respond in a way to optimally produce a protective antibody response. All right, now before we get into this, into the nuts and bolts of what we've done here, I wanted to go big picture and just focus on some of the things that really made this a challenging project. When I initially kind of started pitching this around and trying to get funding to go after this, people said, you know, this is not going to work. There's too many complicated parts here. And there are a lot of complicated parts, and it does make it very challenging. But as you'll see, we got it to work quite well. So what are some of the complications? So I've already mentioned this a little bit. Antibodies can really come in two kind of major forms here. They can be bound to certain cell subtypes, naive cells, memory cells, other types of B cells, or they can be secreted. So we wanted to make sure with our engineering, we didn't mess with the portion that dictates this. So the portion that's bound to the membrane or not bound to the membrane. We wanted the cell to be able to figure that out on its own. So realistically, we only really wanted to engineer the antigen binding portion, so the variable region of the antibodies up near this end. Now, this is further complicated by the fact that an antibody is made from two gene products. So there is an antibody heavy chain. This is the longer piece of the antibody here that can connect to the membrane in this particular example, and also an antibody light chain. So if we wanted to replace the antigen binding portion, we actually have to have an approach that will replace two portions on two different genes. So that makes it a bit more complicated. Now, just to talk a little bit about the genetics here and what we're talking about, the variable portions genetically are referred to as the VDJ for the heavy chain allele and the VJ of the light chain allele. These refer to the gene segments that come together to form this antigen binding portion. So this sounds, I guess maybe on this cartoon, maybe it's not so complicated because it's just two segments. We know what they are. We know where they are. That's not 100% sure or certain, which I'll get into in a moment. But even just to replace these two segments is actually quite complicated. So why is this complicated? Now this hopefully is not an overwhelming slide with lots of different information on it, but I wanted to get in a little bit into VDJ recombination. And this is how each individual B cell during development generates its unique antibody. And so if you look at a heavy chain loci in the genome, in any cell in your body, what you'll find is that there are hundreds of different V segments, dozens of D segments, and then a handful of J segments. VDJ recombination is where each individual B cell first links together a D and a J segment, essentially genetically engineers itself to bring these two together, cutting out all the DNA in between. It then links the V to the DJ segment, again, cutting out all the DNA in between. Every single B cell as it develops does this independently. And this creates quite a challenge for us because normally in a genetic engineering project, we would look, okay, we want to replace this VDJ section here. So let's cut it out. Let's cut to the left of it. Let's cut to the right of it, and we'll remove it and replace it. But we don't know which V's and which J's each cell is going to choose. One cell will choose this one, this one, this one, and the other hundreds. And this can span over a region of about a megabase of DNA. So we don't exactly know where this segment is going to be, creating a lot of challenges from an engineering perspective. Now, I've only talked about the heavy chain loci. A similar process is going on at the light chain as well. There's no D segments involved there, but the V and the J come together in that context. So again, now we're trying to target two different genetic loci with genetic engineering, and we don't know exactly where things are sitting. So we decided to come up with a simpler approach that in a lot of ways, we kind of cheated the system. Here, we started thinking about this locus and, what can we count on? What is going to be the same in every single cell? So what we came up with is using CRISPR-Cas9 to cut DNA past the final J segment So we know this region is going to be intact in all cells. And before the downstream stuff that's important, there's an enhancer that tries gene expression downstream of this. There are the constant regions, of course, downstream of this. But we knew this region would be intact. So the basic idea that we came up with was to cut this region and then insert a new cassette encoding our antibodies of interest, specifically just these end portions that we were getting at before. So in this cassette, it starts with a new heavy chain promoter. Then it has the VJ, so the variable segment of that light chain, that is the part that contacts the antigen of interest. The constant region from the light chain, so it's a full light chain gene in our cassette, a linker between that segment, and then just the VDJ of the heavy chain, so just the variable section of the heavy chain. And what we were hoping to do is insert this in a way that it's downstream of all the variable stuff that we can't really predict, but has its own promoter, so that this promoter would be inserted, and using some of the enhancers and things that are downstream, it would drive expression of our cassette and produce antibodies that, in comparison to the native antibody, which I showed before, are what we call engineered monoclonal antibodies, our emAb B cells. And this, again, will have the variable region of the light chain in light blue. The dark gray is the constant region of the light chain, an orange linker to a dark blue VDJ. So we've replaced this whole antigen binding portion that I mentioned before, but left kind of some of these other parts, the constant region of the heavy chain, which will dictate whether this is bound to a membrane or secreted, left all of this intact. It uses a heavy chain promoter, again, keeping the regulation intact.
Dr. Taylor?
Yes.
You still have the original VDJ. Is that not going to complicate things, or is that something we're moving later?
That's a great question. And I think the idea here, I get this question a lot. And the hypothesis when we started is, if you look upstream of the VDJ that you're using here, there's a promoter. So is that going to drive expression? Is essentially what you're asking, right? And just get some sort of Frankenstein transcript coming out of here. But what we thought is, well, look up further again. There's another promoter. There's another promoter. There's potentially hundreds of promoters upstream of these V regions, and they're just all completely ignored. And what we hypothesized is that these enhancers are all downstream, and they reach up and just essentially the first promoter they find is what they end up driving. That was the theory, of course, when we did this. And there was some evidence that if you inserted another gene into pretty much the same locus during development, it completely shut down B cell development by having something in this region. So that was our hypothesis and evidence at the time that this would actually work. But you're pointing out a good point. This was risky when we tried this. This didn't necessarily have to work. And we thought of other strategies where we wouldn't put a promoter here. We would have something that just uses this natural promoter that's being used. But this, as you'll see, ended up working out quite well. Okay. All right. So as I mentioned, this was kind of a risky idea of what we were trying to do. Engineering of B cells with CRISPR had not really been done when we started this project. So where do we start? And what we started with is not with primary B cells, but we used a cell line called RAMOS. This is a human B cell line that's immortal, will keep proliferating in vitro, essentially indefinitely. And so we designed guide RNAs to target our region. There are lots of different prediction algorithms. We use this one called CRISPR Gold. Essentially, you put in a stretch of DNA. So we put in our stretch of DNA in here, and it spit out potential guide RNA sequences that should cut this region in human, and we also did in mouse B cells, very efficiently, and should also have few off-target cutting effects in other locations. So our first experiment was to take RAMOS B cells. We then electroporated them with a guide RNA bound to Cas9. A few days later, we look for what's called indels. And this is a cartoon example of what these are. That's short for insertions or deletions. So if you just cut a stretch of DNA, it repairs, but it's messy. So you can get small insertions or small deletions in this area. And using just standard Sanger sequencing of this area put into an algorithm called ICE, we can read out the number of insertions and deletions within our population of cells. When you've chosen a good guide RNA, and generally this algorithm gives us great ones. I think they've all worked, everything that we've tried. There's some variation from guide RNA to guide RNA. The one we move forward with here cut about 70% of the DNA sequences, resulting in insertion or deletion. So really efficient cutting in here. And that's what you want when you're trying to do a project like this. So this worked really well in a cell line, and anyone that's ever worked with cell lines and then primary cells can tell you that it doesn't always translate. What might work well in these cell lines might not work well once you get into primary cells. Oh, actually, before I get there, let's go to the next step. So I've shown that I'm able to cut the DNA where I want to cut it. Can I insert the DNA for the antibody and show that I can replace the antibody of interest? So here we did our cutting using guide RNA and Cas9, and then expose the cells to an adeno-associated virus, an AAV, that carries our emAb, our antibody cassette, the genetic cassette that we want to insert into the genome in this region. After 2 or 3 days, we then look for the expression of this antibody. And we can do that in a lot of ways. The easiest for us to look at is using flow cytometry. So here, what we've done is with control cells, these are just cells that go through the process but don't get the DNA for the antibody. We look for the ability to bind RSV. And in this experiment, we've put in an RSV specific antibody. We also look for binding of streptactin, which binds to the linker that I mentioned was between the light chain and the heavy chain in this construct. You can see control cells, basically nothing binds as expected. But when we do our engineering, now about 1/3 of these RAMOS B cells will bind the antigen of interest and have this tag linked between the light chain and the heavy chain. Now, keep in mind, there's no selection or anything like this on the cells. So we've truly engineered about 1/3 in this particular experiment. So does this work with primary cells? So here we purified human B cells from the peripheral blood. We put them into culture for a couple of days and stimulated them with a cocktail of factors that's really designed to induce these cells to proliferate. For these types of genetic engineering and insertions, cell division proliferation is really required to get efficient integration of the gene we're trying to put in there. So we stimulate the cells, get them to start entering cell cycle, then we electroporate with Cas9 in our guide RNA, then expose the cells to this AAV that carries our cassette that we're trying to insert into the cells. 2 days after that, we again look for binding of the antigen that the antibody we're putting in should bind, and this tag is the second level to look at this. And you can see in our human primary B cells, again, this worked really remarkably well in that I think about 1/3 of the cells on average will express our antibody of interest, again, with no selection at all. This is just our engineering efficiency. And when I started this project, I originally wrote in the grant, if we could get 1 to 10% of the cells to express our antibody of interest, I'm going to be thrilled. Maybe I didn't use that exact language, but I think it came across in the grant. And so when the people in my lab, Howell and Carson, and this particular experiment was done by Julia, came to me and were showing these percentages, I think we got as high, if we look at the summary data here, is of about 60% of the cells engineered to express our antibody of interest. So on the bottom, this is just a summary. These are individual experiments where we engineered cells with an RSV specific antibody, an influenza specific antibody, an EBV specific antibody, or an HIV specific antibody. And you can see this worked remarkably well with different antibodies that we've tried to put in. There are other ways you can analyze these types of insertions. You can, of course, use PCR, look for your cassette being inserted into the proper locus. Flow cytometry is an easy way for us to really visualize this and quantitate it quite rapidly. Another way you can analyze this, and I think was important for these experiments, let's make sure we haven't screwed up the cells. Maybe now they can't secrete this new engineered antibody that we've put in. So here we engineered primary human B cells, we stimulated them further, and then looked in the culture supernatant to just confirm that we could detect secreted antibody. And you can see in red, when we've engineered them for an RSV, flu, EBV, or HIV specific antibody, we can detect that antibody in the culture supernatant, and the controls don't have any of those specific antibodies. Now, I think the question that was brought up during the cartoon is a really important one. Do we actually replace the existing antibody? Maybe we get some expression, but there's mixing and matching going on here, and there's something else wrong with the system. So to do this, to get a little bit into the weeds of the different types of antibodies, what we started with in this experiment, again, is primary human B cells. And we selected B cells that had a lambda light chain. So B cells can either express a lambda or a kappa light chain. And so we focused on the cells that just started off expressing naturally a lambda light chain, used our engineering to express an antibody that was a kappa light chain, and looked for the loss of the lambda light chain. If we've completely eliminated lambda expression, even though we haven't done anything to the light chain locus, then it tells us that we've eliminated expression of the endogenous heavy chain. So here, just to orient you to the graph here, we're gating on B cells that either were engineered to express an influenza-specific antibody and then could bind a flu antigen, or the ones in that same culture that didn't express our antibody of interest. We then looked for expression of this lambda light chain. In the cells that didn't get our engineered antibody, pretty much all the cells express a lambda light chain. When we put in our influenza antibody and focused on the cells that were expressing it, we were actually a little concerned when we first looked at this graph, because we noticed that while most of the cells expressing our antibody of interest downregulated lambda, so no longer express the endogenous receptor, there was about 40% that maintained this, suggesting that these cells were actually expressing two antibodies or mixing and matching some of these chains, even though we tried really hard to avoid that. This was further confusing when we looked back at our data from the cell line. I mentioned before the RAMOS B cell line that we had initially started these experiments on. That also expresses a lambda light chain. And when we had done those experiments, it looked beautiful, where on top, the cells that weren't engineered to express an RSV specific antibody were all lambda positive. When we expressed our RSV antibody in these cells, it completely eliminated lambda expression. Now, what I didn't understand at the time, when we were first working with RAMOS cells, is this is actually a peculiarity of this particular cell line. It turns out that RAMOS B cells, unlike normal B cells, don't have the normal chromosomes that B cells have. In the heavy chain chromosome on one allele, there was a translocation that essentially eliminates it. So these B cells only have one heavy chain loci, not the complementary chromosome, whereas a primary cell has both. So this actually suggested that in our cells, we can put in our engineered antibody into the heavy chain that's been utilized, or the one that's actually not utilized by the cell, the second chromosome that's just along for the ride. Now, in some ways, this is bad because we wanted to replace the endogenous antibody. I showed that as our initial goal for doing this, but we decided to use it as an opportunity. If B cells that are engineered can express two antibodies, can we take advantage of that? So in this experiment, we decided to try to purposely create what we called two emAb B cells. Typically, a B cell expresses one antibody, but can we get it to express two antibodies that we will? So we did an experiment where we engineered different populations of cells to express one antibody, then just cultured them as a control, or we put in the DNA for two different antibodies with the idea that one would get on one heavy chain loci, another would get into the other heavy chain loci at some probability. Some would end up with two of the same thing. They would flip-flop which one they were in. Some would only get one. But we wondered if we could force cells to express two antibodies. This again worked remarkably well, whereas our control, the singly engineered, you don't see a lot of cells that can bind RSV and influenza when you engineer the cells independently to produce a flu-specific or an RSV-specific antibody. Whereas when we double engineer, we got about 6% of the cells that actually express both of these antibodies. Now, one thing you might be wondering is, well, why would you want to do this? Why would you want a cell to have more than two antibodies or more than one antibody? And the reason that we're really excited about this potential is there are a lot of situations where a single antibody just isn't enough for what you want to do. At the beginning of this presentation, I mentioned HIV. And trying to develop a situation where you could engineer cells to produce broadly neutralizing antibodies. But it's really well established that even the best HIV broadly neutralizing antibody can't neutralize every single strain. I think that's becoming true with SARS-CoV-2, where we had antibodies that were great at neutralizing early variants, but have lost the ability to bind later. So potentially having cells that could have your primary antibody and then maybe your variant-proof antibody all within the same cell could be incredibly useful.
Dr. Taylor?
Yep.
This seems like it'd be more applicable to a T cell as opposed to a B cell, or more useful in a T cell environment.
Why do you say that?
You can target two different epitopes at the same time to activate your T cell.
Yeah, I haven't thought that much about the T cell approaches. I mean, it should be fairly applicable. I imagine the same rules would be in place. I mean, I could see it being useful in both contexts. Yeah. All right, now, I think a very important kind of proof of principle for this type of approach is to show that these cells can actually be functional and protective. It's great that we can engineer these in culture, but let's take the next step. So for that, we switched to the mouse system and did pretty much a similar approach where we purified B cells, stimulated them, engineered them, selected those cells that were binding of antigen of interest. In this case, it's RSV again. We then put those back into culture with a feeder cell line and some cytokines aimed at stimulating the proliferation and differentiation of these engineered cells to get to the plasma cells and the memory cells that I mentioned before, and then put these back into different animals. Now, at the time of transfer, I just want to talk a little bit about the phenotype of cells. This is flow cytometry again, and here we're looking at two molecules expressed by B cells. CD19 is a hallmark B cell marker. So when we started these cultures, everything would have been in this top left quadrant. This 11% would have been 100% of the cells that went in at this stage. We're trying to push these cells to differentiate into antibody secreting cells. As they do that, cells downregulate the expression of CD19, and many of them upregulate the expression of CD138. So this is what we're really trying to drive here. But we got a mixture of cells that did that and some that didn't. So we thought this was a good starting population to put into our recipient animals. So we engineer, we differentiate, and then we transfer 5 to 15 million of our engineered B cells into a recipient animal. In this experiment, we put these into wild type animals, then waited 7 days and infected them with RSV, looked 5 days later to see if our cells were able to control this virus. On the left here, we're looking in the lungs, we're looking for viral titers after infection. If we don't transfer any cells, we can detect nice levels of virus in these lungs. If we transfer control B cells that didn't get the antibody, but went through the whole process and transfer as well, zero control by these B cells. Our engineered cells are here in red. You see this really crushes down this infection. to the level similar to if you were to take the same antibody and inject it into the animals the day before we do these infections. Excitingly, we've carried these out, particularly in animals that are immunocompromised. As I mentioned earlier, we think that could be a real area where an approach like this could be very useful. So we've done similar experiments, the similar time point, transferring into RAG knockout animals, which lack both T cells and B cells and are highly immunocompromised. In those RAG knockouts, we've also waited a lot longer. So in this experiment, we just waited, transferred the cells, waited for 82 days, then we challenged, and again, protection was maintained that far out. We haven't gone longer. We have some evidence, which maybe I'll show in a minute, that this might diminish after that time point. Checking my time here. Let's skip that here. So I mentioned this ideal approach before. This is easy and efficient. We can engineer cells in 4 days, 3 or 4 days, depending on the species. We can eliminate existing antibody. This has a caveat, is that only seems to eliminate if we get it into the right chromosome that's being expressed. I think that's an important point to keep in mind moving forward. As far as we can tell, we get normal expression and regulation of these antibodies. They can class switch. When on the surface, they can signal normally. And we've demonstrated that they're protective when vaccination is not an option. Now, I don't want to oversell things, so I always want to show at least one thing that didn't work as well as we wanted. In fact, most of the project worked better than I had ever imagined. But there are a few things we need to work on for these moving forward. So before I showed you the protection that lasted out to day 82, but here I want to show you the serum titers of antibody in these animals. So what was initially expected is in red here, this is the levels of antibody we could detect in the serum after we transferred our B cells. This is 6 days after. You can see this is lower levels than if you just injected the antibody at this day 0 time point. But injecting the antibody in black has a pretty well-established half-life and decays over time. Whereas we were really excited at this time point when the lines had crossed, where our engineered cells were maintaining levels of antibody beyond just an antibody injection, which I think is a very important benchmark for an approach like this. Ultimately, we want long-term protection that's completely maintained forever. After day 40 or so here, we start to see a decline in the antibody levels that are in the serum. Now we've looked out at this time point, day 87 or so. We can still find the cells in the animals. We need to more sensibly characterize what's going on. Are the cells secreting antibodies dying off or downregulating how much antibody they're producing? And how can we prevent that? So it's one of the areas that we're really looking forward to exploring in the coming years.
Dr. Taylor, is that affinity maturation affecting the long-term viability of those transformed cells?
Yes, in this particular experiment, we don't anticipate that there'd be any affinity maturation. These are going into RAG knockout animals, so we don't think there'd be any kind of germinal center response. There are no T cells to help with that. So we don't think that would be coming into play in this particular situation. Now, in wild type animals, that could potentially be an important thing to look at, whether affinity maturation is actually having a bad effect on our cells.
I was thinking it was killing them off as opposed to actually doing maturation effects.
Yeah, it could. I think it's something that we really need to explore. Yeah. All right. So just as a kind of big picture summary, where are we and the field going? What are the potential clinical and experimental uses for an approach like this? There are several other groups in the field. We haven't done much of this, but they've explored engineering the cells and then directly transferring them into an animal, followed by a vaccination. And the idea here is that this bypasses this differentiation step that I talked about before. I mentioned and just showed the decline of serum antibodies. Well, maybe that's because we haven't really differentiated the ideal plasma cell that we want that's going to stick around and crank out these antibodies forever. So maybe a way around that is to transfer these cells, vaccinate, and let the immune system, respond in the way that it normally should to generate these subsets. And I think this is a really exciting kind of approach. And the papers that have come out have shown that this is really promising. They can generate these long-term subsets. Germinal center responses look fairly normal in these in these experiments. So it's really exciting about the potential of being able to do that. Now, another idea in the field, and actually I think the first clinical use will probably be in this realm, where there are several companies out there that are really interested in taking advantage of this remarkable secretion capability of plasma cells, not for antibodies, but for other proteins of interest. So I think there are two companies whose primary focus is on enzyme replacement therapies. Instead of injecting this antibody every other week in some cases, what if we could get a plasma cell to just crank out a ton of this enzyme stably for long periods of time? We're working on this a little bit. We'll skip over that slide as well. Just for time reasons, I don't want to take up too much of all of your time, but there are lots of different approaches where you could harness the capability of plasma cells to secrete antibodies to secrete other things. Another thing we're working on in the lab is to simplify things on the other end. Instead of starting with a naive B cell and engineering that, then differentiating, let's start with these subsets that already have the functional properties that we want. What if we directly engineered memory B cells? Perhaps they would survive and function ideally compared to something we made in culture. Same thing goes with antibody secreting plasma cells. This is a particularly challenging target because they're not readily accessible in circulation. They're not proliferating, and that often is required. But there are some ways to get around that we're working on with a collaborator. And I think ultimately, one of the things that I'm most excited about, and myself and a collaborator just got some funding to explore, is let's go completely to the next level and not take the cells out at all. Let's use these approaches to engineer cells in the body. So give essentially a vaccine-like injection, but carrying our CRISPR payload targeted to the cells that we want, whether they be naive or memory or plasma cells, and engineer it, this immune response without this extra step of taking the cells out of the body. So hopefully we'll have some cool results with that in the coming years. So to summarize what I've talked about today, hopefully I've convinced you that we have an approach for rapid and efficient engineering of B cells to produce pretty much any antibody of interest. We can then use these cells to protect against infection. And I think you could transfer this basic idea to any other function or any other context where B cells might be important for oncology implications, things like that. And what might be most of interest to this group is beyond just antibody replacement. Now, I showed you one example of how we could take B cells and make them do something they normally don't do, but could have some uses for us. And in that example, it was to express two antibodies at the same time. But there are other things we can start to do here. Can we further engineer these antibodies to have additional properties that we like? Bispecifics, trispecific antibodies. Could we change their functional capabilities by starting to modify the constant regions in these cells? So I think there's a lot of exciting potential for what we're doing there. Just to end, again, I want to highlight my lab. I've done none of the experiments that I've talked about here. My group really took some big picture ideas that were risky and improved upon them and ran with them and really made these projects what they are. We're also looking to expand the team. I mentioned some funding to, in particular, explore the in vivo engineering areas. So we're looking to fill a couple positions on those projects. With that, I'm happy to take any additional questions and feel free to reach out through email if you have anything after today that you think of or on Twitter as well. And with that, happy to take any additional questions.
Well, thank you very much, Dr. Taylor. We have a question in the chat, And for anybody else that has questions, you can either raise your hand or put it in the chat, and I will make sure that to share that. So the first question here is from Linnea Fletcher, who asked, what about immunocompromised individuals who are advised not to get vaccines? For example, people who have chronic COVID infections and are generating variants.
Yes, I think that's a great question. And I think for a technique like this, it could be a really powerful way to protect these individuals. Now, it depends on the specific reason why they're immunocompromised, how you would go about doing that. Do they have B cells that just don't function properly? And could we correct that in some way, shape, or form, and or provide the antibody that we need? Will we need to get B cells from a different source to be able to do this? Yeah, I think that's a great question. Something that hopefully 5-10 years down the line, we're able to help some people that can't generate these things on their own.
Okay, and Todd.
I guess, well, I'll frame this question. So one is, what I really enjoyed about this was, at the beginning you talked about, well, there's, the experts didn't believe this could work. And I think that's a challenge in science. As the more expertise you develop, the more cynical you become. And there was a naivete here that said, let's do it anyway, which I think is a great message for students thinking about their futures. Any thoughts on that? And then the second is on the RSV. You see the titers drop, and RSV is just a chronically hard thing to vaccinate. And is there any link to the biology? Antibodies that lose titer may not boost, or cells that lose titer may not boost. Yeah.
I think RSV is where we actually set this project up from the get-go, because historically it's one of the ones where generating a vaccine has been challenging. I think there's some exciting things on the horizon there that hopefully cure that problem. Like, we're not trying to replace vaccination in any way, shape, or form. I think vaccination, when it works, is great. We're just trying to create an option for situations where it's not great. And who knows how well those vaccines, if they do work, will work in some of the target populations that are really susceptible to RSV. Again, immunocompromised, premature infants, and I think older individuals as well are another population that gets hit by RSV really hard, even though it's not talked about quite as much.
Yeah.
But yeah, and I guess to comment on your first comment about a little being a bit naive as you go into these things, I mean, my own perspective is I get most excited about the new things, and trying some stuff. And I've talked about all the parts of this project that worked really well, but there are additional parts that didn't work as well.
Sure.
The overall worked, but as we start pushing the limit on what is possible, we do find a lot of things that don't work, but that's okay, and keeps it exciting in the lab.
Cool.
Okay, Francis.
Thank you, Justin, for a great talk. I have two questions. So have you looked at the not the rag knockouts, but the wild type mice? Have you looked at the mouse antibodies against your construct? Because that's a big problem in humans for ha-has. And then the second question is more thinking about the application into humans. And that's the turnaround time. That's the turnaround time, which you may not be able to speak too much to in humans, but how's about in mouse? Because it takes a while for the vaccination to make a difference, right? To actually protect you. And if you could get the B cells in faster, that would at least kick it off.
Yes. So your first question is, I think both your questions are really good ones. The first one, do you essentially generate an immune response to the cells? Because we're putting in this construct that has undergone affinity maturation and also has this linker construct. I think a lot of people are concerned about that being immunogenic, and now you generate antibodies to your antibody and just eliminate all your cells. The short answer is we have not looked very carefully for those types of responses. We used a linker that was developed to minimize some of these things. And I think it might be problematic, especially giving a large bolus of cells all at once. You might be forcing kind of more immune responses. So yeah, a bit of a hand-waving answer to that. I think it's a concern that we need to think really hard about and analyze for and figure out if we can get around if we see it. But we don't know how big a problem that might be. Second part of the question, how quickly are we able to see a protective response after our transfer? Let's see, in the mouse, we engineer, we transfer the cells. That's a 10-day process. The earliest we've looked for antibody, I think, was day 4, and it was in the microgram per mil range, which is the protective range. So if we could do something similar in humans, the in vitro part is pretty similar for humans. I think the tough part there would really be scaling up to the same level of cells. We put in 5 to 15 million cells into mice. What does that translate to in humans? And can we create that many cells? Or would we have to start from a lower baseline and maybe using a boosting type approach in some way, shape, or form to get things to the level we need them to be at? Great questions. I don't have more specific answers than that, especially for the human.
Miko.
Hello. Oh, here we go. Hi, from San Francisco. This may be a little bit of a broad question and maybe even a little tangential. Sorry.
That's fine. Go for it.
Still a little early in my career. I'm maybe a bit naive. I know you mentioned this platform was really designed with viruses in mind, but would there be any applications for this platform in things like cancer? Like, if so, would it work with something like liquid cancers or even solid cancers? I know you're like engineering like the variable regions, so would that be able to be like switched out for maybe something cancer specific?
Absolutely. It's a great question and something that's being explored, not just by us, by other groups in the field and companies in the field as well. I think of the initial kind of B-cell engineering companies that have formed, I think the 2 of them primary focus is on maybe enzyme replacement. Another 2 or maybe 4, we'll see how many companies there are in the next few years, are actually focused on oncology. Whether it's to change the variable regions to target a tumor antigen, for example, or there are lots of other situations where antibodies are used as therapeutics. And we can swap in any of those domains into this, the binding domains into this platform as a way to potentially provide antibody differently. And I think in those applications, what could be really interesting is, often when you give antibodies, you have to give them every 2 weeks, every month, depending on the antibody. But if you have cells in the body that could respond to the target, they could basically self-boost themselves if that target's there. So it could be a really cool situation. And, obviously this is big picture pie in the sky, but I think there is a lot of potential for doing that. And a lot of groups are hitting upon that. So I think your question actually was not at all naive. You hit the nail on the head.
Well, that sounds really amazing work. Thank you.
Thank you.
Linnea.
Well, first off, I want to really thank you for a fascinating presentation. Really enlightening. And my question is this: I was really fascinated by the secretion rate for antibodies. And I assume that's unbelievably high. And they must have investigated why it can do this. And so I'm really curious about that, because it is an amazing platform for secreting proteins in general.
Yeah, I mean, this goes to a couple studies. I think the initial one was many years ago now, looking at these capabilities. And, just to, I guess, put a little context to that, I gave a number of thousands to tens of thousands of molecules per second. If you look at the number of antibody molecules on a normal B cell, memory cell, it's probably around 200,000 molecules. And this is a well-expressed protein on the surface of the cell. So essentially the cells that are secreting are replicating that in 10 to 100 seconds, right? That amount, but pumping it all out. Now, if you look at a plasma cell, it originally was characterized as something different than a B cell because it looks so different. It has completely changed its architecture. The ER has been expanded in ways that they are protein factories. If you look at most of the expression of what makes a B cell, it's downregulated all that stuff. I mentioned CD19, but you could go through a list of transcription factors. It just turns all this other stuff down and just let's get these antibodies. This is my job, survive, produce antibody. And that's what it seems to do. But I know this information for a long period of time and I'm still stunned by it.
Thank you.
Daniel.
So it really strikes me that you're getting around normal immune system development. So I guess my question is, have you started to think ahead to, so how do you avoid unintended autoimmune diseases or autoimmune responses that you might create? Because you're putting in something that you've engineered. You need to know, like, this is not going to attack the person.
Oh,
So,
yeah. I think the key
sort of-
part of that is making sure we're replacing that endogenous antibody. I think that is the biggest risk for an autoimmune response. Because I think, if you're choosing antibodies up front that don't bind to anything in the human body, things that have been injected into people and are perfectly safe, something like that, I think you get around that particular issue. But if we're not silencing that endogenous antibody, all bets are off. So that's one of my kind of fears when we're thinking about this, is that we could just license something that, there are B cells in all of our bodies right now that produce antibodies that can recognize antigens that our body produces, autoantigens, self-antigens. So they're usually not high affinity. They're usually not a problem, but we don't want to make them a problem.
Right.
Yeah. It's a great question. I obviously have spent a lot of time thinking about this and, being worried about this. You don't want to stumble into a situation like that.
Are there other questions? No, there's been plenty. All right. Well, thank you so much. We really appreciate this. And I want to make a plug too for Shoreline Community College, which is not so far from Fred Hutch. And I know you already have there's already quite a few students from Shoreline there, but I will share the information with them as well. And if you like, we do have a LinkedIn group called Antibody Engineers. Feel free to post job announcements there as well.
Okay, great. Thank you. All right. Well, thanks everyone.
Thanks. I'm going to stop the recording here.